Alright, ladies and gentlemen, this is going to be our next video on mitosis, and specifically looking at binary fission. Let's get started on this next video. So if we're looking at prokaryotic versus eukaryotic cells, well, they're going to divide differently. Prokaryotic cells do not go through this cell cycle. And remember, prokaryotic cells are unicellular organisms without membrane-bound organelles. They go through this process called binary fission. Now, eukaryotic cells use the cell cycle. Just as a reminder, remember these are cells with membrane-bound organelles. They have a nucleus. So cell cycle is really important for eukaryotic cells, and we learn about that process. That's G1, S, G2, and mitosis. But prokaryotic cells go through divisions a little bit differently. So what we're going to do in this video is delve into how prokaryotic cells go through and divide to better understand how eukaryotic cells are more specialized, more complex, and how they divide. So if we're looking at the prokaryotic cell cycle, there is no cell cycle here. It is much simpler. What they do is a process called binary fission, and this is a form of reproduction in which prokaryotes utilize to go through and divide and reproduce. Now, if we look at this, remember they don't have a nucleus and their DNA is much simpler. They're really bacterium, and prokaryotes don't need to go through and do very complex functions. So they only have DNA for really basic survival and basic functions of the body of that prokaryote. Now, the other thing that these prokaryotes are going to have are what we call plasmids. And plasmids are going to be these smaller circular pieces of DNA. They're not in the center of the mass of DNA in that prokaryote. They're just kind of floating around in the cytoplasm, and they're just these small circular pieces of DNA. So as we're going through and looking at the division here, let's ignore the plasmids first and just talk about the DNA. So if we're looking at our prokaryotic organism here, it has a cell membrane, and the blue is going to represent our DNA here. Now, this prokaryote is going to go through, it's going to double the DNA, that strung up mass, and the DNA is going to duplicate, which is going to go through and enable for our division. So what we're going to do is create an indent in the center of the cell membrane. We're going to create this cleavage, and we're going to go through and separate these two bacterium, or these two prokaryotes, into two new cells with this copied strand of DNA. So if we look at this, we have two new cells that have gone through and formed. Both of them have this copy of DNA. And remember that DNA is not in the nucleus, it's just floating around in the cytoplasm. But we've essentially gone through and copied that and gone through a simple division here. So what about the plasmids, and what is the importance of these plasmids? Well, the plasmids, if we're looking at this, the blue is representing our DNA, and the black is going to represent our plasmid circles. When the cell goes through and divides, the DNA is separated into two halves, but the plasmids are going to go through, and they're randomly just going through and dispersed in the cell. So we've been talking about plasmids a lot, but we haven't really talked about what a plasmid is. Well, all a plasmid is a genetic structure in a cell that can replicate independently of the chromosomes, and it's typically just a smaller circular strand of DNA in the cytoplasm. So we have our main DNA in the actual bacterium here, and we also have these little tiny plasmids, or this plasmid here. And this plasmid might have genetic material that makes it favorable for this bacterium to grow in a new environment. So let's say we go through binary fission, and this bacterium is going to go through and split into two new bacterium, and the plasmid, we're just pretending that there's only one. It's unequally exchanged here. Now we collect these two bacterium. We put the bacterium with this plasmid in one petri dish, and we put the bacterium without the plasmid in another petri dish. We're going to put these petri dishes in the oven. We're going to heat them up into what we would consider an inhospitable environment. What we're going to notice is the bacterium with the plasmid, well, they're going to reproduce and create this giant colony. Now, the bacterium without the plasmid goes through and dies, and no colony forms. So what the plasmid was really going through and doing was creating proteins that maybe made a larger shell or a larger protein layer around this bacterium, and it made the bacterium resistant to that inhospitable environment. These plasmids are going to be really, really important because they're going to function for a few things. The DNA only provides basic cell function, whereas the plasmids enable adaptation, and they also enable prokaryotes to rapidly adapt to an environment. So if we're looking at bacterium here, Well, we have the DNA, which we know is going to be passed on to the next generation of bacterium. But if a bacterium can go through and pass on these plasmids are like extra strands of DNA that might help the bacterium in some way. Let's say these plasmids allow these bacterium to be heat resistant to an environment. Well, we can go through and the bacteria can reproduce and create lots and lots and lots of bacterium with these plasmids that allow them to adapt to new environments. So what we're getting here is a process where the plasmids are either randomly distributed through the bacterium, or we get conjugation, where we have an exchange of genetically favorable material among the prokaryotes. It allows the bacterium to adapt rapidly to new environments. And this is the reason that most likely we see these bacterium in these Yellowstone hot pools here. So Yellowstone is basically a valley on top of a volcano, and we see all of these really colorful pools of bacterium here. And these pools have bacterium that are really, really resistant to these temperatures. And the most likely reason that this is because these plasmids, bacteria went through, exchanged plasmids, or went through and divided with the presence of these plasmids to make them adaptable to this inhospitable environment. Now, this is important for bacteria because they can divide very, very fast. So reproduction depends on how favorable the environment is, and if the bacteria can rapidly evolve to that environment, we can get lots of bacteria in unfavorable environments based on these plasmids. So as a general rule of thumb, bacteria are going to divide once every 20 minutes. So if we were to just let bacteria go for 6 hours and divide, we would get 250,000 bacterium. There's a lot of bacterium over the course of 1 6-hour span here. So these plasmids are really just extra pieces of DNA that are allowing these bacteria to evolve and adapt to what we would consider inhospitable environments and allow them to reproduce faster and really evolve and adapt to environments quicker.